Good evening, everybody. Tonight's lecture might be of interest to you if your vision isn't as good as it could be, because our three lecturers tonight, their topics, of course, is vision on eye health, focus on eye health. So tonight we're going to enjoy presentations from Dr. Julie Lim, from Joyce Mathin and Professor Trevor Sherwin, who are all experts in their own field on eyes and vision. So for those of you that don't know me, my name's Sue Brewster and I am the Executive Director of Auckland Medical Research Foundation, and I give you a warm welcome for coming tonight and thank you very much. I'm sure what you're about to hear is going to be some amazing research that is being done and the advances in medicine that are happening. So most of you will know that we fund medical research. That is our whole purpose for being. And I think for Dr. Julie Lim, we have funded 5 projects over the last 10 years. And this has really helped her progress her research into her area. Joyce Mathin has had a doctoral scholarship awarded to her last year, which is great. And Professor Trevor Sherwin has had his fair share of funding over the years as well. So, and of course, it's our wonderful donors and supporters who make that all possible. So what you will hear and listen to tonight is really made up from your support and your donations. And because we're lucky enough to have our administration costs funded by a benefactor, 100% of all donations goes directly into medical research. And that's quite a unique position for a charity to be able to stand up and say. So I feel very privileged to be here tonight. By the way, in your packs tonight, you will see there is a feedback form. We'd love it if you give us feedback. It's always really interesting for us to know what you think of the lectures and what you might like to hear about. So without further ado, I'm going to ask Dr. Julie Lim to come up, and she is actually going to introduce the topics tonight and our speakers. Thank you very much.
So, thank you to the AMRF for hosting this event, and thank you so much all for coming. It's great to see so many faces here. So my name is Julie Lim, and I'm going to be one of the presenters tonight. But before we start, I just wanted to give you a bit of an introduction and sort of set the scene for you a little bit and get you to watch a video of what it's like for people living with vision loss.
Can't get onto the bus, you can't find the bus you want. There's just so many things, it's just difficult to describe how often you need sighted assistance or you need guiding.
I didn't get out of the house for 2 years, just stuck in the house.
When you're blind and you get into a shop and you've turned around several times and bought several things, I swear that they'd move the door when you tried to come out.
Couldn't really accept using a cane. I suppose the image of having a cane and just accepting becoming blind.
As my sight went, I just lost all confidence and I couldn't go out really. And then my partner said, you know, you're really going to have to learn, you know, to get out and about because you can't stay indoors all your life.
So I hope you can see from that video the profound impact that vision loss has on a person's quality of life. That vision loss can affect people from all walks of life, and it can affect people who are young and people who are older. Now, some of those conditions you may have heard of before, some you may not be familiar with, but they affect the different tissues of the eye. So before we start with the talks, I'd just like to give you a very brief overview of the different tissues of the eye and how they actually work together to allow us to see an image, allow us to see. So if we look at the anatomy of the eye, and in the front are 2 very important tissues, the cornea and the lens. And they are clear and transparent, and their role is to refract or bend light to a single point at the back of the eye, which is called the retina. And the retina contains very special cells that allow the light to be detected and for this light energy to be then converted into electrical signals. These electrical signals are then transmitted through to the optic nerve and then received by the brain. And the brain is able to encode these electrical messages to allow us to see an image. Now, you would have noticed that that video was from the UK. And so what I want to talk about tonight is how these eye conditions affect New Zealanders and how many people in New Zealand are affected by these conditions. So if we look here at the back of the eye, at the retina and the optic nerve, there are a few very important eye conditions that affect New Zealanders. One of the first is called age-related macular degeneration. And age is a major risk factor. And it affects the macula, or the centre of your retina, so you lose central vision. And it's estimated that around about 200,000 people in New Zealand live with this condition. Another condition is called diabetic retinopathy. About 330,000 Kiwis have diabetes, and this number is increasing. And about a quarter of these people will go on to develop diabetic retinopathy, so around about 80,000 people in New Zealand. And what it results in is damage to the blood vessels in your retina, and so someone who has diabetic retinopathy will view the world like this. The optic nerve also has a condition called glaucoma, and this affects around about 70,000 people in New Zealand. And if not treated, it will result in loss of peripheral vision. So it's like looking through a tunnel. If we then move to the front of the eye, if you recall, the cornea and the lens were really important for refracting or bending light. There are refractive conditions associated with these tissues. They're not diseases as such, it's just that your eyes can't focus properly. So with the cornea, some common conditions that you may have heard of are myopia or nearsightedness, hyperopia or farsightedness, or astigmatism. And although these can be managed through wearing glasses or contact lenses, remarkably, there are around about 80,000 people in New Zealand with uncorrected refractive errors. In the lens, an age-related condition is called presbyopia, and this typically affects people over the age of 40, which accounts to around about 1.4 million people in New Zealand. Now, what happens here is that normally the lens is able to accommodate. It can actually change shape. It can become more rounded or more flatter to allow you to see close up or far away. Unfortunately, as we age, our lens becomes less flexible. Just like as we get older, our muscles and joints get stiffer, so too does our lens. So it's not able to change shape as well. And in presbyopia, what we find is that people can't see near objects, but they seem to be okay with distance. And that's why you might see a middle-aged person reading the newspaper like this. Now, unfortunately, our arms don't get longer as we age, so we have to wear glasses to correct for this. Now, in these series of talks tonight, we're going to talk about tissues in the front of the eye, the cornea and the lens. And our first speaker will be Joyce Mathan, and she'll be talking about the cornea and a condition called keratoconus. And what happens in keratoconus is that your cornea has a more cone-like shape. And she's going to talk about this condition in children with Down syndrome. So then we're going to move from young vision to older vision, and I'm going to be talking next about a common age-related disease in the lens called cataracts. Now, cataracts is the leading cause of blindness in the world, and it's been estimated that there are around about 370,000 people in New Zealand living with cataracts. And what happens here is that your normally clear and transparent lens becomes cloudy, so you can't see. And I'm going to be talking about some of the research that we've been doing to understand this process a little bit better. And then our final speaker is Professor Trevor Sherwin, And he's going to be talking about turning back the clock and how we can use stem cells to potentially regenerate new cells and new tissues, and maybe one day in the future help prevent these debilitating eye conditions. So without further ado, I'm going to introduce Joyce to come and talk about her research.
Thank you for the introduction, Julie. Hi everyone, my name is Joyce Mathan. I'm an optometrist and doctoral candidate in the Department of Ophthalmology at the University of Auckland. In this presentation I will discuss keratoconus and Down syndrome, and I wish to thank the Auckland Medical Research Foundation for providing me with the doctoral scholarship. So the cornea is the front transparent structure of the eye. This transparent structure arises from the highly ordered arrangement of collagen fibres in the cornea. It is important that the cornea remains transparent so that light can pass into the eye. It's located in front of the coloured part of the eye, the iris, and it forms 2/3 of the optical power of the eye. The remaining power comes from the lens. Keratoconus is thought to affect 0.05% of the general population in New Zealand. Instead of the cornea being shaped like a normal dome, it progressively forms a peak. Keratoconus is characterised by increasing corneal curvature, corneal thinning, biomechanical weakness. It can be detected by corneal topography maps. So much like contour maps of landscapes, corneal topographies map the symmetry, the steepness, the regularity of the corneal surface. The exact cause for keratoconus is unknown, but is believed to be multifactorial, including genetic and environmental factors. Keratoconus begins in the early teenage years and may progress to about the 3rd, 4th decade of life, where it may stabilise. It is associated with hay fever, asthma, eczema, and eye rubbing. Initially, keratoconus may be managed with spectacles and contact lenses. In advanced disease, intervention by corneal transplantation may be required. This is a highly invasive procedure. Globally, New Zealand has among the highest proportions of corneal transplants for keratoconus in both general and paediatric populations. This may be avoided by a less invasive procedure known as corneal collagen cross-linking. Corneal collagen cross-linking is a minor surgical procedure. It involves the use of riboflavin eye drops and UVA light. This strengthens the corneal collagen bonds, making it less prone to further progression. It is more effective when implemented in early stages. Down syndrome is most commonly caused by a standard trisomy of chromosome 21. It affects 50 to 80 live births annually in New Zealand and is associated with intellectual disability, visual impairment, hearing impairment, heart, respiratory, and musculoskeletal disorders. International estimates for keratoconus and Down syndrome ranges from 0 to 71%. The range of prevalence values may be due to the age of the participants, the sample size of participants, the ethnic distribution of the participants, as well as the method of diagnosing keratoconus. Individuals with Down syndrome have also been reported to have steeper, thinner, and weaker corneas than individuals without the syndrome. They are also reported to be disproportionately affected by an advanced form of keratoconus. This is known as corneal hydrops. This is a sight-threatening and painful stage of keratoconus. In November 2017, our team investigated the prevalence of keratoconus among athletes with Down syndrome at the New Zealand National Summer Games held in Wellington. The author specifically provided corneal topographies for individuals with Down syndrome, so we obtained the topographies as well as data on athlete demographics. We used 2 different quantitative criteria for diagnosing keratoconus, as there has been no established quantitative criteria for diagnosing keratoconus in this group. So the corneal topography maps that met either quantitative criteria were then assessed by 3 independent eye specialists. Keratoconus was confirmed when it was identified by at least 2 of the 3 specialists. So 110 athletes with Down syndrome were screened. 98 were included in analyses, most were male and most were European. The mean age of our participants was 30 years old. So keratoconus was detected to affect 10 and 38% of participants by the first and second methods respectively. There were similar proportions of bilateral cases detected by each method. Here are some topographies obtained. The warmer colours indicate areas of steepening, and the cooler colours, areas of flattening. Most participants were in less severe stages of keratoconus. So this pilot study identified that keratoconus may be over 700 times more prevalent among individuals with Down syndrome. This highlights a very real need for a designated screening and treatment pathway for keratoconus in this group. This led to the KIND 21 project. The KIND 21 project consists of 3 sub-studies. The first study is a prospective observational study looking at the prevalence of keratoconus in this group as well as other parameters. The second study is an audit of clinical records. The third study is a prospective observational study looking at the outcomes of corneal collagen cross-linking. The aim of our first study is to determine the prevalence, severity, and also the nature of progression of keratoconus in this group. We aim to reach 400 individuals with Down syndrome and provide a thorough corneal assessment for them. The project period for the initial eye exam is expected to end in February 2020, and the follow-ups are expected to end in February 2021. Anyone aged 10 years and older is invited to participate in this study. Study 2 is an audit of clinical records. Here we aim to determine the prevalence, severity, and management outcomes of keratoconus in Down syndrome among individuals with Down syndrome attending the ophthalmology service at Auckland District Health Board. We aim to review records over the last 10 years. This project is estimated to end in May 2020. In our third study, we aim to determine the clinical outcomes of corneal collagen cross-linking in this group. We also aim to create a multidisciplinary team to optimise current corneal collagen cross-linking protocols so that the best clinical outcomes may be achieved. This project is estimated to end in February 2021. In this study, we aim to observe the post-op outcomes over a 6-month period. We will also record specifics about the treatment that was provided, such as whether the type of anaesthetic used, whether it was general anaesthetic or local anaesthetic, and the specifics about the technique. This will also reveal practical challenges of treatment. The multidisciplinary team, consisting of eye care practitioners, developmental paediatricians, anaesthetists, representatives of the participants and their caregivers, will provide recommendations that may optimise cross-linking protocols for this group. Our team have been working very closely with the New Zealand Special Olympics, the New Zealand Down Syndrome Association, the Auckland Down Syndrome Association, as well as others. We were at the National Summer Games in 2017, and following that, our team provided a regional screening of keratoconus at one of the smaller Special Olympics events. More recently, we attended the Auckland Down Syndrome Association Buddy Walk, where we set up a stall and had the chance to meet individuals with Down syndrome and their families and talk about the project, as well as answer any questions they had about the study. We would very much appreciate it if you knew anyone with Down syndrome, if you would let them know about the opportunity to participate in our study. I've got posters available, so if anyone wants some posters to hand out, do get in touch. I wish to thank my supervisors, Professor Charles McGee, Professor Deepika Patel, Dr. Jay Meyer, Dr. Akhilesh Gokul, and Dr. Samantha Simkin for their support, and we wish to extend our special thanks to the many individuals that have been involved in this study. Thank you very much.
So I'm really excited to be here to talk about my favourite tissue of the eye, which is the lens. So the lens is actually a remarkable tissue because it's able to remain clear and transparent over many decades of life. But unfortunately, as we get older, the lens loses its transparency and we develop a cataract. And what our research is trying to do is understand the molecular mechanisms involved in this process so that we can identify strategies to delay the onset of cataracts. So just to remind you where the lens is, it sits here, right behind the cornea, so right in the front part of the eye. And you can see that because of its positioning, it's very exposed to the outside environment. So it's really exposed to things like UV radiation or sunlight, which we know over time causes oxidative damage to the lens here. Now, the role of the lens is to focus light onto the retina. And to do this, it has to be clear and transparent. And this allows us to see a nice, clear, sharp image. Unfortunately, as we get older, we develop a cataract, a clouding of the lens, and so now you can see the light rays scatter, and what we end up seeing is a blurry image, like looking through a dirty, fuzzy window. Now, there are different types of cataract, but the most common form of cataracts is called a nuclear cataract. And this is when the cataract appears in the nucleus or the very centre of the lens. So what I'm going to show you are some images from human donor lenses to show you how this cataract forms. So what we have here is a donor lens that's a clear and transparent lens. It is a pale yellow colour, and that's normal. So our lenses are a pale yellow colour to begin with, and then as we age, the coloration increases, so they become more yellow. But what you can see here is you can see the lines here formed from this grid behind the lens. So the lens is acting like a biological glass. If you have a look at this next image, this is the start of a nuclear cataract, and you might be able to see the appearance of this slight opacification right here in the centre of the lens. This here is what a typical nuclear cataract looks like. You can see that there's browning in the very centre, and so sometimes this is called a brunescent cataract or a brown cataract. And then this is a very advanced stage of cataract, and you can see now that the cataract has actually spread to the outer regions of the lens, and you can't see the grid lines now behind the lens. So it's no longer acting as a biological glass. So we know that age is a major risk factor for cataracts. And if we have a look at this graph, we can see that over the age of 50, the incidence of cataract markedly increases. Now, there is an effective way to treat cataracts, and that is by cataract surgery. So here an ophthalmologist will remove the cataractus lens and replace it with an artificial intraocular lens. And some of you in the audience may have had this procedure before. My mother's just had cataracts removed in both eyes, and so I know firsthand how effective this surgery is in restoring vision. So then you might ask yourself, well, if there's an effective treatment, why do we need a way to prevent or delay cataracts? Well, the reason is the demand for cataract surgery is increasing. Cataract surgery is the most commonly performed surgical procedure in New Zealand, and last year alone there were around about 30,000 cataract surgeries performed. The reason why it's increasing is because we're living longer. Our life expectancy has increased, and this has resulted in an increasing ageing population. And remember, age is a major risk factor for nuclear cataracts. Also, we have an increasing diabetic population, and some of these individuals will go on to form a diabetic cortical cataract at much younger ages, around about 40 to 50 years of age. Now, this type of cataract is different to a nuclear cataract, but the treatment is the same, and that is by cataract surgery. So you can see that because of our increasing ageing population and our increasing diabetic population, this is placing huge pressures on our hospital system. In fact, there are lengthy wait times for cataract surgery in the public health system. If you meet a certain threshold, we are told that you wait no more than 4 months. But many people don't meet this threshold, and so they are waiting much longer to receive cataract surgery. And you may have read in the Herald just on Monday the story about an elderly lady in Cambridge who didn't meet the threshold for cataract surgery, couldn't see out of her eye that had a cataract, and couldn't drive. And the poor lady felt so embarrassed to have to rely on her neighbours to get her milk that she resorted to buying milk powder. So that just shows you the impact of cataracts and how that can really affect your day-to-day living. It can create a loss of independence, maybe a lack of confidence in driving. It can lead to depression and social isolation, particularly if you don't have any family support. There's also strong evidence that there's an increased risk of falls and fractures associated with cataracts. So not only is there an emotional impact, but there's also an economic loss to the individual and to the public health system. Now, because cataract is primarily a disease of old age, it has been predicted that if we can delay the onset of cataracts by 10 years, we could reduce the need for surgery by as much as half. And that would significantly reduce the cataract burden on our hospitals, but also improve the quality of life, particularly of our elderly population. And so it has become our vision in our lab to try and identify strategies to delay the onset of cataracts and reduce the burden of age-related cataracts in New Zealand. So what do we know about age-related nuclear cataracts? Well, we know that in a clear and transparent lens, we have very high levels of an antioxidant called glutathione. It's higher than any other tissue in our body. And it's made up of 3 amino acids: glutamate, cysteine, and glycine. And this cysteine residue is very important because it has this special SH group or thiol group, and it gives glutathione its antioxidant properties. What it does is it donates electrons to reactive oxygen species so that they can't then go on and inflict oxidative damage to the proteins. And what we know is that when we age, our glutathione levels decrease in the lens, but they don't decrease homogeneously, they decrease specifically in the lens nucleus. And this is shown really beautifully by images obtained from Gus Gray from a laboratory. He's developed this imaging technique that produces volcano plots. And what these plots are showing are the levels of glutathione as we age. So each of these volcano plots represents a human donor lens. We've got a young donor here at 29, a middle-aged donor here at about 51 years of age, and our oldest donor was 82 years of age. Now, if you have a look at the rim of these volcanoes, they're filled with warm colours like yellow, and yellow means very high levels of glutathione. And what you can see that with all these donors, even with increasing age, they all have that yellow rim, which means their glutathione levels remain at high levels. But if you have a look at the craters now, they're a little bit different as we age. You can see that these warm colours here are now replaced with more blue and purples. And what this represents is a depletion of glutathione, specifically in the lens centre or the lens nucleus. Now, what we know is that when these glutathiones decrease to a certain level, around about 1 millimolar, it triggers a series of events. And these events include protein oxidation, loss of soluble proteins, protein aggregation, and cataract formation. Now, if you look at this flowchart, you might think, oh, it must be quite easy to cure cataracts. Surely you just add more glutathione. Why don't you just restore the glutathione levels and you'll prevent cataracts? But unfortunately, it's not as simple as that. And many people have tried glutathione supplementation and antioxidant supplementation, and none of these studies have shown to be effective in preventing or delaying the onset of cataracts. And we believe because it's due to a lack of understanding of how antioxidants are actually delivered to the centre of the lens. And this understanding is based on lens structure and physiology. So I'm going to give you a quick overview of what we know about lens structure and physiology. So if we have a look here, this is the schematic, and here you can see the lens suspended within the eye. If we take a close-up look and then we cut this lens from the top to the bottom, we'll see a section that looks like this. This is a typical cross-section of a human lens. What you'll see in the blue line that totally encapsulate the lens here is the capsule, and that gives the lens support. Underneath the capsule are these epithelial cells in yellow, and they line the front part of the lens. And then at a region called the equator, these epithelial cells, they differentiate to form fibre cells, and these fibre cells form the bulk of the lens. Now you'll notice that they're colour-coded, and that's because these fibre cells are laid down at different stages of development. In the embryo, as a fetus, as a juvenile, and as an adult. So these cells here in the centre of your lens are the oldest cells in your body. They were laid down in utero. And as the lens grows throughout life, new fibre cells are packed in amongst these older fibre cells. So you can think of the cells of the lens a bit like rings on a tree trunk, where the older cells are in the middle and the younger cells are on the outside. Now, if you recall, the role of the lens was to transmit that light to the back of the eye, the retina. And so, in order to do that, the lens has developed some features that enable it to do this really, really well. One of these features is the loss of cellular organelles. So if you have a look at these epithelial cells and these very outer fibre cells, they contain these grey little dots, and they represent cellular organelles such as nuclei and mitochondria. And what that means is that these cells can make new proteins. Now, these organelles are degraded or lost in these nuclear layers because having these organelles would actually scatter the light rather than allow the light to be transmitted through. But this poses a major problem for cells in the nucleus because they don't have the machinery to make new proteins. So the proteins in this area, they have to be long-lived because there's no way to replace them. And that's what makes this area of the lens susceptible to oxidative damage and the formation of a nuclear cataract. The other feature is that the lens is avascular. It doesn't contain a blood supply because the heme pigments in blood would absorb the light rather than allow the light to travel through. But once again, this poses another problem for these cells in the nucleus because in the absence of a blood supply, how are these cells going to obtain antioxidants? Well, over many decades of research, it's been shown that the lens operates what's called a microcirculation system. And this is driven by a circulating current of sodium ions. So it enters at the poles and exits at the equator. And as sodium enters the lens at the poles, it draws in water and antioxidants to be delivered deep, deep down into these central fibre cells. And these fibre cells then express transporters that help to then accumulate these antioxidants to protect them from oxidative damage. So why is this so important? Well, it is our hypothesis that with increasing age, there is a failure of the microcirculation system to not only deliver those antioxidants deep into the lens, but also a reduced ability of antioxidants to then be accumulated within these cells. And as a result, we believe this is the reason why there is a depletion of antioxidants specifically in the lens nucleus and nuclear cataract formation. So what our research is trying to do is understand this pathway a little bit better. What are the antioxidants accumulated in this area, and how are they accumulated in this area? And my work has focused on an antioxidant called cysteine. And you will be familiar with cysteine because it's one of the amino acids used to make glutathione. Remember, it gives glutathione its antioxidant properties. Now, I'm focusing on cysteine for a couple of reasons. First of all, when you supplement with glutathione, glutathione actually becomes a target for proteases, so it's broken down really fast. So it has very low bioavailability, whereas cysteine doesn't have that problem. The other thing is that cysteine is much smaller than glutathione, and so delivering something much smaller into the centre of the lens is going to be a lot easier than delivering something bigger. Now, when you read the literature, cysteine is actually transported around the body as cysteine. And cysteine is just simply two of these cysteine residues that are linked together by this special bond. In other tissues, what people have shown is that cysteine is taken up by a cell and then rapidly converted into cysteine. And the transporter that does this is called the cysteine glutamate antiporter, or CGAP. So cysteine is brought into the cell, it's rapidly reduced to cysteine, and in other tissues it can be used to make glutathione, the antioxidant. And what we wanted to know for our research was whether CGAP was present in lenses, and specifically, was it present in the lens nucleus? And could this be a way where we could try and enhance cysteine levels in the lens nucleus to protect from oxidative stress? So to do this, we used a technique called immunohistochemistry, and we were really lucky to obtain human donor lenses from the New Zealand National Eye Bank. So what we did is we took donor lenses, and this time we actually cut them through the equator. And this is what we expected to see. So this time now you can see your capsule, which is the blue line, the epithelials, which are the yellow cells, and then the fibre cells. And you can see the beautiful architecture of these fibre cells, how they are geometrically arranged. Now, we used a high-powered microscope to visualise these cells, and this was the first time that anyone had actually done this. Using this technique, what we can see are the cells in the outer cortex. This is the capsule, this is the epithelium, and these are your young fibre cells. These are the cells that were laid down when we're an adult, when we're a juvenile, when we're a fetus, and these are the oldest cells in your body. They were laid down in embryo. And you can see how beautiful these cells are, but also how different they look in the different layers of the lens. Now, using this technique, we were able to localise CGAP and see where it was in the lens. So what you've got in red is just the outline, and what I'm going to show you in green is the labelling for CGAP. So what we can see is CGAP is present in the outer cortex and in all the nuclear layers. So CGAP is expressed in the outer cortex and the nuclear layers. We've done a lot more work on this, but what we've proposed are two roles for CGAP. The first is that in the outer cortex, these cells can make new proteins. They can make new antioxidants. So we think that CGAP in this region can be used to bring in cysteine. It can be converted into cysteine, and this cysteine used to make glutathione. But if you recall, in the nucleus, this region can't make new proteins. It can't make new antioxidants. So we think the reason that CGAP is there is to bring in cysteine, reduced to cysteine, and potentially this used as a means of providing an antioxidant to the lens nucleus. Now, this was an exciting find for us, and so now we're actively investigating whether CGAP can be targeted to enhance cysteine levels in the lens nucleus and protect from cataract. So where do I see ourselves in the future? Well, at the moment, cataract surgery is the most effective way to restore vision, but the demand for cataract surgery has created an increased need to develop alternative strategies. And our long-term goal is to develop an eye drop that can be used to delay the onset of cataracts. We don't think that there will be a single magic compound that will do this. We do think it will be a combination of factors. So, for example, if CGAP can be used to deliver more cysteine, can we be sure that it will be converted into cysteine? Because it's as cysteine that it is effective as an antioxidant. And what about the microcirculation system? Can we use factors that can upregulate the system and enhance the delivery of cysteine into the centre of the lens? And then long term, if we want to think about translating this into a clinical setting, we need to identify a therapeutic window in which cysteine supplementation may be beneficial. In conclusion, oral antioxidant supplementation has been an area of intense therapeutic investigation to delay cataracts. But unfortunately, they have proven to have no effects in protecting against cataracts or reducing the progression of cataracts. And we believe that this is due to a lack of understanding of lens structure and function. If you recall, the lens is avascular. It doesn't have a blood supply. So it's going to be very difficult for an oral antioxidant supplement to get to the lens, let alone the centre of the lens. And we think that with our knowledge of lens structure and physiology, we can potentially harness the existing machinery of the lens to increase the delivery and the accumulation of antioxidants in the lens nucleus to delay the onset of cataract. So finally, I want to thank all the funding agencies who have supported me over the years, but in particular, the Auckland Medical Research Foundation. I really wouldn't have been able to do this work if it wasn't for their continued support. And then finally, this is of course a team effort, and I really must thank the director of the lab and my research mentor, Professor Paul Donaldson, the other members of the lab, Gus Gray, Renita Martis, Ivy Lee and Louis Knight. Also the clinicians from the ophthalmology department, Professor Charles McGee and Professor Deepika Patel in particular. And of course, special thanks to the families of donors and the New Zealand National Eye Bank that have been able to use this really valuable research to advance our understanding into cataracts. Thank you.
So we're going to finish off this session today with a bit on turning back the clock, and so it's the promise of regenerative medicine. You'll notice from the two speakers who spoke before me that I'm probably the most in need of some regenerative medicine. What is regenerative medicine? Well, basically, if you have a heart attack or a stroke, then the heart cells or the brain cells that you've lost during that event are never going to be recovered. You're never going to get them back unless we can use regenerative medicine to try and replace those cells in the tissues that have been affected by the adverse event. We like to think that we're probably one of the most sophisticated animals on the face of this planet, but in terms of regenerative medicine, we are not. The simple newt, salamander, and the axolotls have got it over us. Here you see a newt that has lost its leg, but over a period of months, it is able to regrow that limb, the bones within the limb, the vessels, the muscles, and restore that limb back to where it once was. And you now see this little newt waving around its It's nice regenerated limb, and he's quite happy. And what's the worst thing we can do to him now? We can take it off again, but that's to let you see what happens. Firstly, the tissue repairs itself. It's got to close the wound site. But then if we look inside the limb, you'll see what happens. And we used to think that stem cells got transported into the area and then regenerate the tissues, but that's not what happens. The cells flood out of the bone, out of the muscle, out of the skin, and reform new bone, new muscles, new skin, and reform that limb. So that the cells that were in the area where the injury occurred reprogrammed themselves and became more like the stem cells. So what is a stem cell? You'll hear a lot about stem cells in the news. It's a single cell that can replicate many, 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 many times. Lots of cells can replicate a few times, but a stem cell has to be able to do it lots and lots. It's got to replace your cells for a whole lifetime. But it also needs to be able to give rise to different types of cells. So it needs to be able to form a heart cell or a kidney cell or a brain cell or a cell within your eye. Where do we get these stem cells from? Well, the most powerful stem cell on the planet is a newly fertilized egg. So that fertilized egg is a single cell which can grow a whole new human being. But there are also embryonic stem cells. So in the first few divisions of that egg, you will retain some stem cells within the embryo. Once the embryo develops into a fetus, around about week 9 of development, we call the cells fetal stem cells. But of course, these stem cells have huge ethical issues around their use. They're coming from aborted embryos or fetuses. So we concentrate in our lab on the stem cells which are either in the cord tissue, the umbilical cord, from a birth, or in the adult stem cells which are present in your tissues. So you'll have some stem cells still within your tissues which regenerate the tissues every day. If we know, people say, why can't we just inject stem cells into us and let them regenerate the whole body? Well, it's not as simple as that. If we transplant stem cells into the body, then they face many issues. Firstly, they need to survive where we put them, and they need to proliferate, so generate more cells. They need to be able to differentiate into the different types of cells that we want them to be able to differentiate into a heart cell, muscle cell, brain cell, or eye cell. And then they need to be able to mature into the cells which form those organs and integrate into those organs properly and function. Beyond that, they also face two dangers. Either our own immune system might react against them, or those cells might mutate and become a tumor within us. So we can't just inject stem cells unless we have done the research and we know how the cells are going to react and how they're going to be integrated into the body. So adult stem cells, we are fortunate, as we are interested in eyes, that there are some really beautiful stem cells within the eye at an area at the edge of the cornea, which Julie talked about earlier, which is called the limbus, and that's where our stem cells reside that reform the front of the eye. We have, if you have a former thing called limbal stem cell deficiency, where we have destroyed those limbal stem cells so we can't reform the eye, then you get a condition which is called limbal stem cell deficiency. This is a patient of Charles McGee's who had a chemical burn to the front of the eye. You can see the cornea is pretty well heavily scarred and right out to the limbus, and we've lost most of those limbal stem cells. With best practice treatment, so that was 55 days after the accident that he had, 265 days after the accident, still heavily scarring. There's no clearing of the cornea. There's no stem cells there to do any restoration of that cornea. Day 609, you're getting up towards, 2 years' time, and you've still got no improvement in the eye whatsoever. The patient can't work. They can't function very well. That eye is effectively blind. And it was at that point that Charles turned to Jane, who's in the audience, and myself, and said, look, we need to do something unusual for this patient. He's not getting any better. We should think about a limbal stem cell expansion. So Charles took him to theatre, took a tiny piece of limbal tissue from the relatively good eye, although he managed to get chemical in both eyes, a tiny piece of tissue, just 1 millimetre squared, and bring it back into the lab and Jane took on the job of growing up those stem cells so that we could then get more stem cells to put back in the eye. You can see after 96 hours in culture, you can see the cells growing out from that tiny piece of tissue. And out at day 20, they've grown to where the blue arrow is. And then this, it's not very high tech, but these tissues grown in a petri dish within the lab with special media, and that is what's supplied back to the surgeon to go back and put it back into the patient. So the patient underwent a double operation with a corneal transplant and with that expanded stem cell transplant over the top. You'll just see the edge of the amnion which is carrying the stem cells out there. So day 819 for that patient. So if you look back at the original 65 days after the original accident, that's how the patient was. After that corneal transplant and limbal stem cell transplant, that's how the patient then presented. A great result for that patient. And we've continued on with that technique, and we've done, we're into double figures now with Charles's patients with chemical burns to the eye. That inspired our further research, because we then want to go and look at those stem cells. Can we refine our techniques? Can we define how best to use these stem cells, how to isolate them from the eye? We're lucky enough that we have within our department the New Zealand National Eye Bank, which collect corneas from cadavers, but supply that tissue to theatre for corneal transplant surgery. The surgeon takes the centre of the corneal tissue out of that corneal button which was supplied to them, but they only use the central part for the transplant, and we get back the rest of it, which includes the limbus, which allows us to then go in and try and isolate the stem cells from that tissue. What we find is when we do isolate them, they form spheres. That's one of the ways which we can identify them. It was when the first stem cells were identified, this ability to form spheres became central to isolating them. Those spheres label with the proteins that we expect them to for stem cells, and we know that they lose those proteins when they start differentiating. So we are pretty convinced that they are good stem cells. What's the capability of those spheres? Well, if you look, there's two spheres on the right-hand side of that picture there, and we use time-lapse video microscopy to look at that, and you'll see the potential of those spheres. So you'll see some cells divide. You see those little cells pop, they go round and then pop into two cells, so that those spheres are producing lots and lots of daughter cells which can then migrate out and repopulate its surroundings. We also found, again with our video time-lapse microscopy, that we saw some unusual features of how these cells migrated out from the spheres, and they migrated out in an orthogonal fashion, so that we got a directed migration, and they left a gap behind that you can see under here, so that they form a structure which was very similar to that which we find in the limbus. So it's almost like in the dish, they are reforming the limbal tissue from which they came. We wondered whether these spheres of limbal stem cells will be able to be put back into the eye and repopulate the tissue themselves. So we took those limbal buttons which I showed you before, and we cut it up into sections, so they've got wedges of corneal tissue. These have been decellularized, so all the cells within them have been taken out, and we then seed them with our stem cell spheres to see whether we can repopulate that tissue. So we go into that tissue, when we put some spheres, just implant them right into the limbus where we think they are needed. You'll see 3 spheres there implanted into that tissue. Then, in order to help us see it, we stain the sphere green, so that any cell which is really live and really healthy labels green. If they turn red, they're dying. But these spheres are really happy, they're really healthy cells, and you can see the sphere implanted at the limbus. So if we shine a blue light on them, they fluoresce green, so that we can just follow the cells once we've implanted them. There's our green spheres implanted into that wedge of corneal tissue, human tissue, in a dish, not in a human being, but in a dish, but still human tissue. And after 7 days, you can see that the cells have migrated out of those spheres and started to repopulate the limbal area. After another week, those cells have migrated further out, right to the edge of the corneal tissue that are left. They don't migrate any further than that. They stay within the corneal tissue, and they don't repopulate the sclera down here, which is the white of the eye. They stay on the cornea. So we're quite happy. Those spheres look like they're going to be a real advance in the way we are able to transplant cells into our patients with limbal stem cell deficiency. There's another type of stem cells which we work on, which I didn't introduce you to earlier. So in 2006, a Japanese scientist, Shinya Yamanaka, who went on to win the Nobel Prize for his work, took some skin cells from a patient and through careful experimentation figured out that we only need to turn on 4 genes in able to push that cell back to being more like an embryonic stem cell. And because they were induced by the genes and because they were able to turn into many types of cells, they were called induced because we induced the genes, and pluripotent because they can produce many types of cells. So they're IPS, induced pluripotent stem cells. And we've made those in our lab from skin cells from patients. And we've been able to work them up in the lab, and they are a very powerful potential tool for putting cells back into the patient to treat any of those diseases which I was talking about earlier. If you take them in the lab, they start forming embryoid bodies, and embryoid means embryo-like. So they start looking a bit like an embryo that we were talking before. What we're also able to do is to induce them to start forming neural crest cells. And neural crest cells are just a type of cell which is expressed early on in development, which will go on to produce things like the central nervous system, but also the major cells that form the front of the eye, the cornea. So we induce them down into the neural crest cells. We take those neural crest cells and we show them another factor to induce them, and you can see these AP2A spheres down here. That's our spheres. There are embryo bodies which are producing the cells, and then we can switch on the proteins which are part of our corneal cells, the Cataracts protein here. So we can induce them down the right line to repopulate our tissue. So we did exactly the same experiment. If we take a wedge of corneal tissue and instead now we seed them with our induced pluripotent stem cells, what happens? And you see exactly the same result. You can see the spheres of the tissue down here, and the cells that have migrated out from that tissue have gone onto the corneal tissue, gone as far as they can within the corneal tissue, and then stop. And they don't repopulate the sclera, the white of the eye down here. So they've done exactly the same thing. So they're looking pretty good as well. So I hope in that brief introduction, this was an image which was drawn by an artist many years ago for a science fiction article, which looked at how you might use these balls of stem cells in the future to regenerate human tissue. It's not a big jump to make when this is one of our spheres of human tissue that goes back into that tissue. It looks pretty similar to the artist's impression, but it's now science fact rather than science fiction. We have to be very careful though. Again, as I said, we can't just go putting stem cells into a human being and letting them just run riot without having done the research to know what is going to happen to those cells. It's too dangerous. You can't just put them into a patient. So the Lancet Commission, commissioned in October 2017 on stem cells and regenerative medicine. For those of you who don't know, the Lancet is one of the oldest medical journals in the world. And it's very, very highly regarded, and so comes out with these commission papers to look at how we are going on with the use of stem cells and regenerative medicine. And it found some worrying things. So there was the growth of poorly regulated clinics who were setting themselves up for stem cell treatments. They were appealing to desperate patients and families who had no other alternatives. But they also found that it was being fed by people's expectations that new therapies should become available quicker than they are. But without the research, it's a dangerous road to take. And where are these poorly regulated clinics, you might ask yourself? Are they in China or India or some of those countries where they might not have the same ethical regulations that we have? No. this report came out in the New England Journal of Medicine, which looked at the treatment of patients in the United States at an unregulated clinic. And these patients were given stem cells which were extracted from their fat. So from their own fat in their bodies, their own stem cells, and then they just injected those cells into the vitreous of the eye, so just in front of the retina. And those patients who were desperate enough to go for the treatment because their vision was poor, those patients who were 20/30 at the beginning ended up at 2200. And those patients who were at 2200, 20/20 is good vision, yeah? So those patients who were 2200 at the beginning and very, very poor vision ended up almost blind, light perception only. So they can see, they can tell if you've put the light on, but they can't tell if anything's moving in front of them. So absolutely dreadful way to conduct a stem cell clinic. There were telltale signs when you looked at the clinic where they were treated. The clinic had no research track record. They hadn't done the background work to enable them to give those stem cells to patients in a regulated way. The patients were told they were joining a clinical trial, but they were charged for the treatment. You would never charge a patient to go on a clinical trial. And worst of all things you could do, they treated both eyes at once. So an experimental treatment, and they treated both eyes, and they've ended up blinding the patients in both eyes. I hope I've convinced you that we are making progress in stem cell research. We need to be careful about how we use it. There are some people who are out there using it unregulated and without the research behind them to teach us how to use it properly, but that's where we come in, that's where the AMRF come in funding the research to enable to put those treatments in place, but in an ethical way.
Well, thank you very much to our three presenters, and from that last presentation I learned a few things. One is that a newt can regrow its own limbs. Two is if you're charged for a clinical trial, it means that it is unethical and you shouldn't be involved. And of course the last one was that scientists do have a sense of humour. So that's really great to see. But all three presentations were fantastic, and we all would have learnt some things along the way. So I'm just going to ask our three presenters to come back up the front here, please. And we're going to do question and answers for those people that have them.
How useful are tissue donors of an older age group as compared with those who are younger?
All tissue was really greatly received, but we can learn a lot from old but also young donor tissue. What we know is that with age there are certain modifications that occur with age, and then there are certain modifications that occur with cataracts, and you can only know that from using older donor tissue, so it is really valuable for our research.
The other thing I should say there is some of the research we've been doing recently, which I wasn't able to show you, shows us that there is a difference between stem cells obtained from male tissue and from female tissue. And the stem cells remain more viable and more powerful for longer in female tissue.
Good evening. Cysteine and cysteine. Why is it, I must have missed something, why is it necessary to transition from cysteine to cysteine? Why not just go straight with the cysteine?
Yeah, it would seem like a much more simpler system, but in fact, when you look at what's transported around the body in the plasma and in the ocular fluids, you find a lot more of cysteine, the oxidized form, so it's much more stable. And so the lens is actually very clever. It knows that it can take up this molecule. It's an oxidized compound, so it seems counterintuitive to want to take up something oxidized, but it has the capacity to actually convert it into something really useful. So it's a much more stable form. It can be transported by the body, and tissues have a transport mechanism to actually help with that accumulation for cysteine.
Dr. Lynn mentioned, of course, that age is a major risk factor with cataracts. Are there any correlations with eye colour and or with exposure to the sun?
That's a really good question. We know that UV radiation can cause significant damage over time. So if you wear sunglasses that are able to filter the UV light, I think that helps prevent your risk. We know that UVA can actually penetrate quite deep into the eye and actually reach the lens. I'm not sure about eye colour. I'm not sure if one of the clinicians know about eye colour and whether that plays a role as well. But I do know that UV radiation in sunlight does cause significant oxidative stress to the lens. So wearing sunglasses will help reduce the risk of that. But obviously there are other factors involved as well. Charles, would you know about eye colour?
Most people have thought that blue eyes are more predisposed, but I don't think there's actually any good evidence. But for blue eyes and pale skinned people are more prone to sun damage, but it probably doesn't make much difference in terms of your cataract, because if your eyes are blue, behind the blueness there's actually a black layer cutting out the light. So everyone, in an evolutionary sense, has a black layer keeping the light out from their iris.
Trevor, just a question again about age. In terms of donating corneas for clinical use, is there an age that you can, it's a cut-off for donating them for a corneal transplant in the clinic?
There is, but it's actually quite high. So we accept tissue for transplant into patients. So the eye bank collects the tissue and processes it, and they will continue to collect eye tissue up to 85 years of age.
So I'll make the cut?
Sorry?
I'll make the cut?
You make the cut by a long way. It's perfectly good tissue. We take it in the lab. We test it for the viability of particularly the endothelial layer, which is very fragile, and look for groups of dead cells and whatever. But lots and lots of 85-year-old tissue is very, very usable, very viable.
You showed the picture of the damaged cornea after stem cell improvement. What was the improvement in the vision?
So it would have been 6/12. In 20/20 vision, that would be 20/40. The vision would be significant to allow them to drive. Yeah.
Given that age is a factor in cataracts, and we get our children to wear sun hats and things to stop them from getting sunburnt and developing skin cancer, should we also be encouraging them to wear sunglasses from an early age?
I think it probably would be a good idea, because those UV rays can actually cause a lot of damage over time. I mean, there are other risk factors as well, so smoking, environmental pollutants. There also may be genetic factors or systemic diseases as well that contribute. But yeah, UV radiation is a major risk factor.
What is the risk level for those of us who have had our cataracts done and have got beautiful new lenses? Is there a risk factor of getting cataracts when you have new lenses in your eyes, when you've had both as many of us have had cataracts done?
Well, there is a small chance of getting a secondary cataract, and I believe it's because some of those epithelial cells, those yellow cells that I showed in the lens, are left behind. And over time, what can happen is that they can undergo what we call an epithelial mesenchymal transition. And they can actually grow and actually hinder the pathway of light. So it is a potential complication that can happen as a result of cataract surgery.
Can I ask a little bit around funding? If you're constantly researching but also looking for money, if you don't have that fund, does everything have to be put on hold until you can fund your next part of research?
Yes, I mean, I've been funded on what we call soft money for many years, so my salary is supported by funding. So I do spend a lot of my time trying to write grants, and we've been fortunate as a lab group to be able to get by, but it is getting harder and harder because it is a really competitive process. But we've just been really fortunate to be able to have charitable trusts like the AMRF to keep us going, but it is difficult.
So when you get to my stage of the career, I don't work in the lab anymore. So the youngsters work in the lab, and part of my job is spent writing grant proposals to raise funding to keep the research going in the lab and to fund their positions within the lab. That's just kind of how the science works.
I'd like to ask, is it Joyce? Sorry? Yeah. On the subject of keratoconus and people with Down syndrome, what's the long-term situation if you are in the position of being a Down syndrome person with keratoconus? Can that be corrected, or does it have to be surgically corrected?
So there's been no studies in the literature at the moment about how keratoconus progresses among individuals with Down syndrome. If it is progressing, what can be done is corneal collagen cross-linking. So if it's detected early, corneal collagen cross-linking is the minor surgical procedure that can strengthen the corneal bonds. So detected early at a young age, you strengthen the corneal bonds at a young age, and that can prevent it from getting worse to a stage where a corneal transplant would be needed.
Okay, it's just that as people get older and older and more Down syndrome people seem to survive longer and longer, it doesn't seem to me that I'm running into Down syndrome people who are actually suffering severe loss of vision. So I just wondered, is there an increasing population of people suffering in this particular way?
I think over the next 3 years we'll get a better idea, especially with the status in New Zealand. And it may also be that individuals with Down syndrome may be less likely to complain about reduced vision. So these things can go undetected because their nature is to tolerate a lot of things. So yeah, hopefully time will tell with this project.
Thank you.
Professor Sherwin took us on a major chemistry tour with a stem cell presentation, which I found particularly fascinating. And I wonder if the possibilities have been addressed of optic nerve thinning as a consequence of glaucoma.
We are using the cornea as a sort of testing ground. We are fully planning to go on and develop some of those stem cells for uses at the back of the eye as well, for photoreceptors or even ganglion cells. So we would need to regenerate the ganglion cells which are lost, but it's perfectly possible in the future. Not close, but we are learning all the time, and those are probably more difficult cells to target, so we're targeting the easy ones first. But as the research develops, there's no reason why.
Thank you. Another one for Joyce, following on from Jenny's question. Is a 21-year-old Down syndrome woman a young person?
Yes. So, basically, keratoconus is known to progress in individuals under 30, and then it tends to stabilise in 30 years old, 40 years old onwards. So if someone with keratoconus at 21 comes in, what we'd do is follow them up at 3 monthly intervals, and what we're looking for is to see if this is progressing. It might be that their corneas are just steep and thinner but stable, but if it is progressing, then we should really consider corneal collagen cross-linking. Also, with individuals with Down syndrome, their corneas start at a lower corneal thickness. With cross-linking, there's a safety limitation for that corneal thickness, so we don't want them to progress beyond the safety limitation of the procedure.
Coming back to the corneal tissue donation for transplants, if one has had cataract surgery with both eyes, does that preclude the use of corneal material?
No. It doesn't. One of the beauties about cataract surgery getting better and more sophisticated is that in doing the cataract surgery, we do less damage to the cornea. So we will damage the cornea a little during corneal surgery, but it's minimised now with more sophisticated phacoemulsification techniques. So there's no reason why that cornea can't then be donated to another person.
Joyce, I've got two, maybe three-part question, just to finish with a bang. Firstly, I know that you're the clinician up there, so this is why it's aimed at you. After cataract surgery, lots of patients come back and they have a laser procedure. Could you explain why that is and what it is?
So what Julie brought up earlier was exactly right. So the epithelium cells grow on the posterior capsular surface of the lens, and what can be done at routine checkups after cataract surgery is to look for that epithelial growth. Laser can be provided. It's called It's called YAG laser capsulotomy, that sort of blasts those epithelial cells to create a really nice space, a clear space in the lens so that light passes through to the retina unimpeded or less impeded.
The other part is about more simple corrections than surgery, such as sunglasses for children. Firstly, how young can a child be to get sunglasses and how accessible are those? And the other one is looking at contact lenses in Down syndrome, because I know other people with keratoconus can use the special hard contact lenses. Why aren't we looking at using that for Down syndrome individuals?
Getting sunglasses as young as possible is always helpful. You can get now spectacle designs that accommodate the flatter nose bridges of babies and younger people, so if they are out outside, I think we should be sun smart and have sunglasses on as much as possible. With regards to contact lenses, yes. Initially, keratoconus can be corrected with spectacle lenses. You get to a point where the spectacle corrected vision isn't so good, and contact lenses tend to do a better job, specifically hard contact lenses. In individuals with Down syndrome, that may be more challenging because that insertion and removal technique takes time to learn. And also, there's a really stringent hygiene protocol around contact lenses to make sure that we don't put them at risk of infections when that's preventable. Having said that though, there is a range of abilities among people with Down syndrome, and they are very high functioning people that can tolerate and do actually very well with hard contact lenses. A participant we saw recently, without contact lenses, he could just see fingers. With contact lenses, he was just below the driving standard. So it makes a huge difference. For some people, contact lenses can make a huge difference to them, even in this group.
And look, just to finish off, as you've heard, none of this would be possible without the funding that is made possible by all of you here who donate to Auckland Medical Research Foundation. We just come into the 21st century. We have a tap to donate on your way out if you feel so inclined. It's a $25 donation, and we're finding that a lot of people do like to give in some way or form. And also in your packs, you'll find other ways that you can fund wonderful work that's going on, like our three presenters have talked here tonight. But most of all, too, I'd like to thank you for coming out tonight, and I hope you found the whole presentations tonight valuable. So thank you, and before I forget, 2nd of July is our next lecture. So I'm just pre-promoting. We're going to trial one during the day because we know in the middle of winter some people find it harder to go out at night. I know I do. And that is going to be on neurosciences, so dementia and Parkinson's disease. So we'll promote that ahead of time as well, so watch this space. And once again, thank you to our wonderful, wonderful presenters.